Okay, well, welcome everybody. I'm Dr. Mike Tranfaglia. I'm the medical director of FRAXA Research Foundation. And this is the latest in the FRAXA webinar series. Today, we're discussing mechanisms and biomarkers of sensory hypersensitivity in the fmr1 knockout mouse. And before we get to our excellent speakers, I would like to just say a word about the format for today's presentations. We will have two speakers talking for approximately 30 minutes each, which will leave us another 30 minutes for questions and answers after both speakers. And as questions arise, feel free to ask them via the chat function, and we'll save up all the questions for the end. So with that having been said, I'd like to introduce our first speaker. Dr. Khalil Razak is a professor of psychology at the University of California at Riverside. And he's going to be talking to you about some of the superb work he's done with the Fragile X mouse model, some of which I'm proud to say has been funded by FRAXA. So with that, I'll turn it over to you, Khalil.
Thank you, Mike. I hope you can all hear me. Let me share my screen. Okay, thanks for the introduction, Mike, and thanks for the invitation, Katie, Mike, and FRAXA to give us the opportunity to present the lab's work in this forum. Good morning to all of you. Hope you're all doing well. So let's get started. I'll make the first half of the presentation, and my collaborator for the last 10 years, Devin Binder, will continue for the next half an hour and be happy to take questions after that. So I want to provide a broad context and rationale for the types of studies that we do in our group here at UCR. And one of the main interests is this association between autism spectrum disorders and sensory processing disorders. As most of you know, 80 to 90% of children diagnosed with autism spectrum disorders also have associated sensory processing issues. In fact, it's so strongly associated, it's now a core diagnostic feature in the latest DSM. So we are broadly interested in what are the underlying mechanisms, like what are the mechanisms that link the autism spectrum disorders with sensory processing disorders, and how to treat the sensory processing disorders and when to treat. What is the time frame? Is there an optimal time frame to treat? And the other major question we're interested in is, can a reduction in sensory issues early in development produce broader behavioral improvements in autism spectrum disorders? And to study this issue, we have been focusing on Fragile X syndrome. And to this audience, I don't suppose I need to give much introduction to Fragile X syndrome, but the lack of FMRP protein because of transcriptional silencing arising from a genetic mutation is thought to be the main reason underlying Fragile X syndrome. And many of the symptoms are listed here. And we are really interested in the association between Fragile X syndrome and sensory hypersensitivity. In particular, we are interested in auditory hypersensitivity in Fragile X syndrome. And there are many reasons, and I'd like to provide the justification for our work in that realm. But first, I wanted to mention it's not just auditory system that has sensory processing issues in Fragile X syndrome. It is seen across the sensory systems, but it is most sort of prevalent and consistent and debilitating in the auditory realm. Humans and children in particular with Fragile X syndrome exhibit sensory hypersensitivity, and this includes very strong and consistent hyperacusis, high sensitivity to normally occurring everyday sounds. The fmr1 knockout mouse, which has been studied for a number of years now as a model for Fragile X syndrome, also shows auditory hypersensitivity. And many of you know that one of the most common phenotypes that is tested in Fragile X syndrome is audiogenic seizures. But even beyond audiogenic seizures, there are many more points of evidences that point to hypersensitivity in the auditory system, which I will talk about in the next few slides. The other major rationale for our interest in the auditory system is that the neural circuits that are involved in sort of early low-level auditory processing are likely to be more conserved across species compared to more complex social and cognitive networks. I mean, what makes us different from a mouse is the fact that we have highly evolved, complex social and cognitive networks. But at the level of basic sensory processing, things are likely to be more similar in terms of circuits as well as in behaviors. And the other major rationale is the fact that Fragile X syndrome is a neurodevelopmental disorder, and there's a rich history of studying development of the sensory systems. And it's also relatively easier to study the development of circuits in the sensory system as opposed to development of circuits involved in social and cognitive sort of behaviors. That's another major rationale for us. And these similarities potentially allows us to engage in translation relevant research by establishing experiments that have strong analogy in the stimulus used, in the analysis used, in the experimental paradigms used between mice and humans. So taken together, we believe that there is increased likelihood of increasing the translational success while understanding the pathophysiology at the cellular and circuit levels by focusing in on the auditory system or sensory processing in general. So in our labs, myself and our collaborators at UCR, we study auditory hypersensitivity. We're interested in cellular and circuit mechanisms, generation of translation relevant biomarkers, and identification of treatment pathways and time points. So in one of the earlier studies that we did in my lab, which is work done by my very first graduate student, Sarah Rothschafer, what we did was we recorded in vivo responses from the auditory cortex of wild type and fmr1 knockout mouse on the FVB background. These were done, at least in the initial part, we were doing this in urethane xylazine anesthetized mice. And these were adult mice, 2 to 5 months old. And what you see in figure panel A is what's known as a post-stimulus time histogram. And each little vertical line here indicates the occurrence of an action potential in response to a sound that was about 10 milliseconds long. It's a 10 millisecond tone that started at 0 milliseconds. So you're looking at the spiking activity of an individual neuron from a wild type mouse or a knockout mouse in response to a very brief tone presented at the same sound level. And what you can immediately see is that in the wild type mouse, when you play a sound, you get a response, and basically the response stops. But in the knockout mouse, what you see is that once you play the same sound, you get a strong onset response just like you got in the wild type mouse, but the response just keeps going on. And that was one of the earliest sort of suggestions of what might underlie auditory hypersensitivity, at least in the knockout mouse. And this is quantification shown across the population. If you measure the number of action potentials produced over 200 milliseconds, so 0 to 200 milliseconds following sound onset, you see that the action potential count is higher in the knockout mouse compared to the wild type mouse. The onset response, which is the response that occurs in the first 50 milliseconds following sound onset, is not different. It is the inability to inhibit these ongoing responses that makes the difference between the wild type and the knockout mouse. Okay. So what this indicated to us was that for the same sound, cortical neurons produce more responses and over a longer duration of time in the fmr1 knockout mouse. In the same study, Sarah Rothschafer also showed that if you measure the frequency tuning curve, which is a measure of the receptive field width of the auditory neuron, the knockout mouse showed individual neurons in the knockout mouse showed broader frequency tuning. So a neuron in the wild type mouse, like this one, has the classical V-shaped tuning curve, which is how many frequencies does the neuron respond to as you change the intensity of the sound. What you see in the knockout mouse is that tuning curve is broader, meaning the individual neurons in the knockout mouse respond to more frequencies than an individual neuron in the wild type mouse. This is quantification of that data, but essentially what it is showing is that across the population, there is a broader tuning. And what that means is that if you play a single tone, more neurons in the auditory cortex of the knockout mouse will respond compared to what happens in the wild type. Meaning, for the same sound, more of the auditory cortex will be activated in the fmr1 knockout mouse. So around this time, we got really interested with these phenotypes in terms of just increased response magnitude and increased tuning width. And around this time, we started interacting with Kim Huber and Jay Gibson at UT Southwestern, who were also interested in sensory hypersensitivity, and they were looking at the somatosensory system. And with Craig Erickson and John Sweeney from Cincinnati, who were also interested in in children with Fragile X syndrome and how sensory hypersensitivity might be an issue there. What are the mechanisms there? So together, we successfully applied for a U54 collaborative research on Fragile X syndrome with sensory sensitivity as the hub that connects the three different groups. And the UT Southwestern group was interested in more of the mechanisms at the level of molecules and cells and networks in using in vitro slices from the knockout mouse. We sort of formed the in vivo hub of this collaboration. And of course, Craig and John were interested in the human side of things. And we used sensory hypersensitivity. And we were interested in addressing the question of what are the mechanisms of abnormal sensory processing in Fragile X syndrome. So the work that I'm going to present to you from now was under the auspices of these collaborative work. And right off the bat, what we wanted to do was make sure that we had stimulus design and experimental paradigms that are analogous between the human work, the mouse in vivo work, and the mouse in vitro work. So we designed a few stimulus paradigms. The first one was habituation, which is simply asking if you keep on playing the same sound, how does the auditory cortex respond? And we did this in humans, and we did this in mice, but we didn't do that, at least at that point, we didn't design to do that in the in vitro slice work. Then we looked at resting EEG, so just basically baseline EEG, and measured the power and asked, are the power distributions abnormal in the EEG responses from humans and mice? And this one we were able to also do in the in vitro slice. Then we started designing stimulus that will address temporal processing, like how well does the underlying neurons or neural mechanisms follow certain changes in time in the stimulus. And we were able to do this in humans as well as in mice. And then the last sort of collaboration was on trying to design the same stimulus paradigms to understand whether stimulus-driven response amplitudes are different in humans and mice, and also in vitro slice preparation, where you can electrically stimulate the slice. So the goal was to use common phenotypes from slice to preclinical to clinical pipeline and test potential therapeutics on these common phenotypes. So here's I'm going to present a sequence of data slides now that were based on the work that we did on the mouse, and then I'll summarize it by showing the similarities between the mouse phenotypes with what we find in the human, or what Craig and John find in the human EEG work. So here's the first study that was conducted by my postdoc, Johnny Lovelace, who showed by recording in the auditory cortex, again in anesthetized mice, and playing the same sound, repeating the same sound again and again. So here's just a noise burst that is repeated 5 times a second, 5 hertz. And then he was measuring event-related potentials, or ERPs, and then asked, what is the amplitude of the major negative going component known as the N1 component, and how does that amplitude change with each repetition of that sound? And that's what you're looking at. And as you can see right away, that in the wild type mouse, the response was strong, and then it very quickly habituated as you kept on repeating the sound. And this is well known in the auditory system literature, is that there is strong habituation of ERP as you keep repeating the sounds at a fast rate. What you see in the knockout mouse is that you also have habituation, but the habituated response sort of asymptotes at a stronger level compared to the wild type mouse. Meaning, for the repetitions of the same sound, the auditory cortex will be activated at an elevated level in Fragile X syndrome. This is showing population data across the recordings that Johnny Lovelace did. This is repetition at 2 hertz. This is repetition at 3 hertz. This is repetition at 4 hertz. In every case, you see habituation in both groups, but always the knockout asymptoted at a higher level than the wild type. Meaning, if the mouse is in a constant stimulation environment, the auditory cortex doesn't ignore the sound as much as the wild type auditory cortex does. And then Johnny, again, our postdoc, transitioned to become more sort of similar, translation relevant with the work that was done in humans, because all the human EEG recordings were being done in awake but relaxed humans. And so he developed a system where we place electrodes on, basically these are epidural electrodes to record EEG. So they are on top, sitting on top, the screw electrodes are sitting on top of the dura, and the mice are awake and freely moving, and we are recording EEG signals. And we record from two electrodes. The FC electrode is the frontal cortex electrode, and the AC electrode is the auditory cortex electrode, with a reference placed in the occipital lobe. And once we get EEG signals like this is called resting EEG signal, because the mouse is not really doing any task, it's not receiving any sounds, nothing, it's just baseline EEG. We can take signals like this and then perform a frequency decomposition using a Fourier analysis, and then estimate the power in each of the different canonical frequency bands, going from very low frequencies, which are in the delta band, to the high frequencies, which are in the gamma band. And it is well known in the field now that these different frequency bands are associated with not just sort of cortical activation, but also maybe associated with specific cognitive functions. And we were interested in seeing if there are any differences in the wild type and the knockout mice in these EEGs. So this is a paper that Johnny published in 2018. And what he found was that the gamma band of frequencies between 30 and 100 hertz showed elevated power in the knockout mouse compared to the wild type mouse. And this is your power spectral density, frequency on the x-axis, and power of the frequencies in the y-axis. And you can see that in both the auditory cortex and the frontal cortex, there is increased gamma band power in the knockout mouse at rest. Okay. So this indicates abnormal oscillation power at rest, and the high frequency power, particularly the really high frequency gamma power, is an indication of elevated spontaneous noisy activity in the absence of any stimulus in both of these different regions in the cortex. This is quantification of such data, and you'll see some examples of this form of data, so let me explain. The y-axis here is a ratio of wild type. So we take the power in the knockout and then divide it by the power in the wild type for each of these frequency bands. So anything that's at 1 means that the power is the same in the wild type and the knockout. Anytime you see values greater than 1, that means the power is higher in the knockout. And anytime you see values less than 1, the power is less in the knockout compared to the wild type. And in fact, when Dr. Binder starts his presentation, you'll see graphs like this, and it uses the same sort of analysis technique. And this is auditory cortex in the top and frontal cortex in the bottom. And you can immediately see that the power in the gamma band is almost twice or more than twice in the knockout compared to the wild type. And this is, again, quantification. We also see increased power in the delta band, but we haven't been able to repeat this more consistently. In every study that we've done in different cohorts of mice and with different people doing the same sort of experiments, we see this consistently happening in the knockout mouse. So the second sort of stimulus, so that the data that I showed you are basically from resting EEGs. There's no stimulus involved. The next sort of stimulus paradigm we used in collaboration with Craig and John Sweeney in Cincinnati was this type of sound known as chirp modulated noise. Okay, what that is or chirp modulated sound. What that is it's a constant sound whose amplitude is modulated. So as you can see, the amplitude of the sound is modulated, but the frequency of that modulation increases over time. Okay, so basically it is an amplitude modulated sound whose frequency of modulation increases. And this sound is useful because this is what is known as a envelope varying or time varying stimulus with frequencies that are commonly present in syllables that we use in speech. So this goes from basically 0 hertz all the way to 100 hertz. Much of the frequencies that are present in naturally occurring sounds, and they occur with the sort of modulations that are present in the in natural speech and other vocalizations that are seen. And then we play the sound, and then we record the EEGs, and you can see there's a strong onset response, and then the response of that is seen in the EEG sort of follows this modulation pattern including all the way up to 100 hertz. So you can see the oscillations in the EEG response. This is data from humans, from John's and Lawrence data that shows this following of this modulation in the response. And then what we can do is measure how well does the response follow the stimulus. And we can use what is known as a phase locking factor and estimate if you play the same sound again and again and again and again, how consistent is the temporal response to the sound across the different stimuli. And using this phase locking factor, this is also known as intertrial phase coherence. There are different ways that people describe this. I might use ITPC or intertrial phase coherence or phase locking factor sort of interchangeably because they're basically showing the same thing. But what this is telling you is giving you an idea of how well is the temporal fidelity of the response maintained to a sound in different groups of animals. This is a method to evaluate temporal processing in the auditory system by entraining the neural oscillations to the modulation frequencies in the sound. We can also do the same sound in reverse, which means it starts with a high frequency modulation and goes down in modulation frequency. We have done both and seen no differences in the response. I'll just show you the upsweep, as we call this, increasing in frequency. Okay, so this is an ITPC plot or the phase locking factor plot, and you can see the frequency of the modulation is present on the y-axis, so going from 0 to 100 hertz. And this is a 2-second sound, so 0 to 2 seconds is on the x-axis. Now you're causing the system to oscillate at the frequencies that are present in the sound. Therefore, the response typically is on a diagonal, which is following the modulation frequencies. So at the beginning, the sound is at low frequencies, so the response is at low frequencies. Towards the end of the sound's at high frequencies, and the response you see are at higher frequencies. And this diagonal, the different colors are showing the ITPC, the temporal fidelity. So the hotter the color, the higher the ITPC, meaning the redder the response, or yellow or red the response, there is greater ITPC, that is better temporal response compared to blue. So as you see in the blue, basically there is no sort of phase locking, but in the diagonal you see strong phase locking. You can take this ITPC measure and then compare between the groups. So here's the wild type response on average, and here's the knockout response on average, and then subtract one from the other to see if there are differences. And that's what we've done. So this is showing you knockout minus wild type. So you take this and subtract this out. And what you can see is that the ITPC, or the phase locking factor, is reduced in the knockout compared to the wild type. So blue indicates less in the knockout. So this indicates reduced fidelity in temporal processing across repeated trials of the same stimulus in the knockout mouse. Okay. And we also did the standard noise burst ERP, or event-related potential. Many of you are probably familiar with this sort of a sound and response. And in fact, this has been very consistently reported in Fragile X syndrome, is that the N1 amplitude on average is larger in humans with Fragile X syndrome compared to typically developing control. And that's exactly what we see in the mouse, where in both the auditory cortex and in the frontal cortex, the N1 amplitude in the knockout mouse is higher compared to the wild type. And this, again, indicates sort of an enhanced synchrony. This is a population response. ERPs are population responses. And typically, a larger amplitude in N1 indicates enhanced synchrony across neurons in the auditory cortex. And the sort of the last phenotype that we discovered in the earlier studies of the Fragile X mouse, and this is consistent with the first data set that I showed you, where if you play a sound, the knockout mouse just continues to respond, whereas the wild type mouse responds and then shuts down its response. And what you're seeing here is what is known as stimulus-induced EEG power. And this is recorded in response to the broadband noise that you saw for ERP. But what you're seeing here is a time frequency plot. So time's on the x-axis and frequency on the y-axis. And you're looking at the power in response to the sound that you played. And you're looking at induced power. And if you pay attention to, say, here, where you have the wild type mouse at P60, this is an adult wild type mouse, and you play a sound, you get an onset response, which is seen here in dark red. And then you have this sort of ongoing response that happens over longer latencies. And this is standard response in the auditory system, where the onset response is followed by what is known as induced response, which is ongoing fluctuations of synaptic activity in the auditory cortex. Now, what you see in the knockout mouse is that the same thing, except it is elevated, is strongly elevated in power, right? So the ongoing activity following a stimulus is very, very high in the knockout mouse compared to the wild type mouse. And this shows you a subtraction, this minus this, and you actually can't see the clusters, but there are dotted lines here showing which parts of these are significantly different between the knockout and the wild type. But just taking a look at this, you can see that the ongoing power is significantly elevated in the knockout mouse. So it indicates ongoing synaptic activity in the cortex following stimulus, and this long latency power is enhanced in Fragile X syndrome. Again, in response to a sound, the cortex doesn't shut down, it just keeps on going. So to summarize quickly, the differences that we see in cortical responses between the knockout and the wild type mouse. So the first major response phenotype is that the single neurons in vivo show hyperresponsiveness to sound. So a given sound causes more response. There's more, the broader excitatory frequency tuning curve. So for a given sound, more neurons will respond. The ERP-N1 amplitude habituation is reduced in the knockout mouse. ERP-N1 amplitude is larger. The resting EEG gamma band power is elevated. And the non-phase locked so-called induced power in response to sound, ongoing induced power is elevated in response to a sound in Fragile X mouse. And then the chirp stimulus showed that there was a reduction in gamma synchrony to temporal modulations in sound. So the way we think about the neural mechanisms of auditory hypersensitivity or cortical hypersensitivity in Fragile X syndrome, at least in the mouse, is that it's a quadruple hit. There are more responses, more neurons responding synchronously, not habituating, and increased levels of background noise. And this is sort of the milieu in which the sound is being processed by cortical circuits in this quadruple hit model. So it's a milieu of enhanced background noise within the auditory cortex, within which stimulus-driven responses need to be generated. Okay, so what is really exciting to us and the collaboration that we have with the U54 group is that almost all of the EEG, ERP phenotypes that we've observed in the mouse are very similar to those observed in humans, not just in terms of magnitude. It's in many different ways. We collaborated in a way that the stimuli and the experimental analysis, all of that was pretty identical, and we see very similar sort of phenotypes. And that's what's really exciting to us in terms of taking this in the translational level. So here's human data on ERP. And again, you can see that there is habituation, but the asymptote is higher in red, which is the Fragile X group compared to typically developing. This is the mouse data that I showed you. This is the mouse EEG data that I showed you. The resting EEG power in the gamma band is elevated in the mouse. Same thing happens in humans. This is data from Cincinnati. And interestingly, when you look at the slice work that was done by Jay Gibson and Kim Huber, you also see an elevated baseline gamma power in the mouse. This is the chirp data that I showed you in the mouse. Knockout minus wild type is blue in the diagonal, which means that the ITPC, or the phase locking, is reduced in the knockout. Same thing is seen in the human when you do the Fragile X minus typically developing subtraction. So this is exciting to us because we can now use the EEG responses to as biomarkers. At least in humans, you can use this as biomarkers to sort of stratify groups for treatment, or to use them as outcome measures in clinical trials, and have a preclinical sort of model to test different avenues of treatment pathways. Okay. And I should mention, this is data from Van der Molen papers who also showed that there is an increased ERP amplitude, which we also see in the mouse. So I'm going to spend maybe another 5 minutes or so, or 5 or 10 minutes, talking about what we think are the underlying mechanisms of some of these abnormal sensory processing in Fragile X syndrome. So this is the data familiar to you, but I'm showing you this because this is a recent paper in which my student Teresa Wen recorded from P14 mice and P21 mice and basically asked the same question of, when you play a sound and look at individual neurons' responses in vivo, what is the nature of the post-stimulus time histogram, right? So you play a sound and you measure the number of action potentials. And what she found was at P14, the wild type and the knockout mice produce rather similar responses. There's no difference in the population between these two, wild type and knockout at P14. But by P21, you see what we see in the adults, in that the wild type produces a response, the neuron produces a response, and then it shuts down, whereas the knockout neuron produces a response and keeps on going. Okay, so the difference is there at P21 between the genotypes, but not at P14. So we sort of think that there's something going on between P14 to P21 where these hypersensitive responses emerge during development. So the question then is, what's happening in that time window between P14 and P21? And in the literature, there are strong suggestions across multiple sensory systems, visual system with work done by Carlos, somatosensory system with work done by Contractor and also Huber's group and Gibson's group, showing that the PV or paravalbumin containing cells, which are inhibitory neurons in the cortex that also express the calcium buffer protein known as paravalbumin. These PV positive neurons are associated with some of the sensory symptoms that are seen in Fragile X mice. So we focused attention on these PV neurons. Interestingly, many of these PV neurons, in fact, the majority of the PV neurons, have what are known as perineuronal nets that surround them. Okay? So PNN or perineuronal nets surround PV cells. So here's an example of the red staining showing you the PV cell, and then the green staining around it is it's a net that forms around these PV cells. And these PNNs typically form preferentially around PV cells, and the majority of PV cells, in many cases, have these PNNs associated with them. And loss of PNNs, and PNNs have been historically studied in terms of development, and there's a lot of classic literature on how PNNs may be involved in the shaping of the critical period window in the visual cortex, and then how loss of PNN may actually be important in opening up plasticity windows and so on. And more recently, we are starting to understand precisely what PNNs are doing to the function of these PV cells. And one of the main outcomes is that the presence of PNNs basically is thought to increase the excitability of PV cells. So PV cells are inhibitory. If they get more excited, then they're going to inhibit more. And loss of PNN in PV cells will reduce that sort of network excitability that is given by the PV cells. Okay. And the PV cells are also well known to be involved in several bands of gamma oscillations. And that's another reason, because we saw changes in gamma oscillations, we also started focusing on PV cells. This was work done in collaboration with Dr. Irina Ethel here at UCR. And we basically, or her lab, stained for PNN and stained for PV and compared at different ages the wild type and the knockout mice. And what she found was between P14 and P21, the PV cell density, PNN cell density, as well as the PV, PNN, co-localized cell density development is abnormal. And it is happening precisely between P14 and P21. It's that time window where we also start seeing the deficits in the electrophysiological responses. This is a paper published by Teresa Wen in 2018. And Irina has worked with this endopeptidase known as matrix metalloproteinase 9, MMP9, for a number of years. And what's really interesting is that one of the targets of MMP9 is the components of PNN. Agrican and other components of PNNs are targeted by the MMP9 for cleavage. So this is an extracellular matrix. The PNN itself is an extracellular matrix, and MMP9 is involved in cleavage of PNNs. So what we hypothesized was perhaps the deficits that we see in the PNN system is because of the increased levels of MMP9 in the Fragile X cortex, right? So MMP9 cleaves PNN components, and as is familiar in the literature now, MMP9 is an FMRP target. So in the absence of FMRP, there's elevated levels of MMP9. And we also have shown, or Irina has shown, that MMP9 is upregulated in the auditory cortex of fmr1 knockout mouse. So here's the data that Irina's group published, is that at different ages, P3, P7, P12, P18, and even going into P40 now, is that the activity of MMP9 is on average elevated in the knockout mouse auditory cortex compared to the wild type mouse. So then the logical next step was asking if reduction of MMP9 in the FMR1 knockout mouse, does it normalize auditory processing? And that was the study that was done, again, by Teresa Wen. And she used a model in which basically it's a heterozygous for MMP9 and full knockout for FMR1. So here's the mouse model that she studied, full knockout FMR1 and heterozygous for MMP9. And she compared that with the typical wild type and the full FMR1 knockout. And what she found for PNN cell density and PV cell density that have PNN, this you've already seen, is the reduction of PNN and PV-PNN in the knockout mouse compared to the wild type mouse. But when she reduced or essentially halved the MMP9 in the FMR1 knockout mouse, there was a recovery. In fact, in some cases, the recovery was even greater than the wild type mouse, that the PNN expression as well as the PV expression came back to wild type or above wild type levels when MMP9 was reduced. And electrophysiologically, also, we saw the effect. Again, here is the familiar post-stimulus time histogram. This is your wild type response from a single neuron. This is a knockout response from a single neuron. And this is the heterozygous response. And you can see that the knockout is higher than the wild type. But then when you remove half of the MMP9 from the system in the knockout, then you bring it back to wild type levels. Okay. So the working hypothesis in our group now is that in the wild type mouse, you have normal levels of FMRP that maintains the MMP9 levels at whatever the normal level is. That affects PNNs around PV cells and maintains a normal EI balance. In the absence of FMRP, there's an increased level or increased activity of MMP9, and that causes the loss of PNNs, which reduces the excitability of PV cells, which then causes an altered EI balance and essentially increased hypersensitivity in the Fragile X mouse. To summarize this part of my talk, cortex is operating in Fragile X is operating in a milieu of enhanced background noise, enhanced sound evoked activity, and reduced habituation. P14 to P21, at least in the mouse, is when hypersensitive responses seem to be developing, and we think of this as a crucial window to target therapeutic approaches. The abnormal development of PV, PNN cells contributes to auditory hypersensitivity, and this has been seen in other systems, including visual as well as somatosensory systems. And targeting MMP9 may restore the EI balance by working on PNNs and the PV cells. And I have the two asterisks here to remind me to acknowledge FRAXA's contribution in two very recent papers. So these are both hot in the press, just came out recently. One using minocycline treatment and showing that the EEG abnormalities that I showed you, at least some of them can be reversed. So there's selectivity and sensitivity can be reversed with minocycline treatment, which is sort of a non-selective drug, but already FDA approved to reduce MMP, which one of the effects it has is to reduce MMP9. And a second paper that was also published by a postdoc that was funded by FRAXA, Patricia Cogram, she showed that using a more selective inhibitor of MMP9 known as SB3CT, we can also obtain reversal of the EEG phenotypes that we've shown. And my collaborator, Devin Binder, will be presenting data now that shows that the EEGs that he's been recording are also sensitive to drug treatments and can be used as biomarkers. So the most exciting part of all of this work over the last 5-6 years has been this collaboration that we've been able to establish with people working on in vitro slices, as well as people working in the human population, to identify similar phenotypes by hypothesizing, by rationalizing that the circuits underlying these phenomena must be relatively more conserved in humans and mice. So I will stop here now. And in fact, I should. So this is the work that I presented were done mostly by FRAXA-funded Jonathan Lovelace, my postdoc, and then my former grad student who's now graduated. And so thank you, FRAXA, for getting us really started on all of this. And then we moved on to NIH and DOD funding on this. So I'll stop here and then I'll let Devin take over, I think.
Well, thank you very much, Khalil. That was fantastic.
Thank you.
And we'll have time for questions at the end of the presentations. So remember that chat function is what we'll use for questions. But in the meantime, without further ado, I'd like to introduce our second speaker, Dr. Devin Binder, who's a professor in biomedical sciences at the School of Medicine at the University of California, Riverside. And Devin is not only a neuroscientist, but he is a neurosurgeon, a practicing neurosurgeon. And he's been involved in the development and application of novel neurophysiological techniques. And I'm proud to say he's also a FRAXA grantee. And he'd like to talk to you about some of the work they've done with multi-electrode arrays and EEG biomarkers in Fragile X syndrome. Devin?
Thank you, Mike. Can you hear me okay?
Sounds good.
Great. Well, welcome everyone. It's really a pleasure and an honor to be involved with FRAXA and also with this presentation today. And I want to acknowledge that Khalil and Irina, my UC Riverside colleagues, a few years ago got me involved in this work, which has just borne incredible fruit for us. So we're very lucky to be involved in this. My background was in epilepsy research and neurophysiology of seizures. So that's another, of course, condition of hyperexcitability, and there's different levels of hyperexcitability, and there's this interesting, overlap and comorbidity of Fragile X patients with epilepsy because of the hyperexcitability phenotypes. But that's sort of my background, and I got involved in order to try to develop new EEG methodologies to try to determine some of these phenotypes in more detail. So without further ado, I'll just move forward. Khalil did a great job sort of explaining the background for this work and how it's been really keyed to the human work done by our colleagues such as Lauren Etheridge and John Sweeney and Craig Erickson, as exemplified in this figure, which basically shows that they found in patients with Fragile X compared to healthy controls that there's this elevated gamma power at the resting state. So that was sort of a huge stimulus to us to try to figure this out in the mouse. And of course, the two electrode studies that Cleo mentioned started this out in our group. The other thing, of course, was that the patients also showed this chirp phenotype, as Cleo mentioned. So I won't go back through this other than to say that we basically started out wanting to reproduce the gamma and chirp phenotypes based upon this work. This was Lauren Etheridge's paper in Molecular Autism in 2017, showing that there was indeed this cortical synchronization to chirp phenotype in the Fragile X patients. So the goals of this research are, of course, stimulated by the human data to develop these measures of both this sensory circuit dysfunction and using neurophysiological methods as such, of course, EEG, and to do that in the Fragile X mouse model. And try to figure out what are the reproducible EEG biomarkers that are reproducible across multiple people doing these studies. And that's an important issue so that there's some reproducibility and in order to basically be able to use these for therapeutic and translational studies. And so we, of course, first wanted to verify or understand, and Cleo's gone through a lot of this work already, do the Fragile X knockout mice have this increased resting gamma power reproducibly and the reduced phase locking to auditory chirp stimuli? And secondly, and more translationally, can we engage these EEG biomarkers with drugs? And in particular, do drugs have effects on various EEG parameters? And then, as I'll come to, the acute versus chronic issue of drug administration. So Johnny, of course, was, and of course, as you heard from Khalil, supported by FRAXA, started with these studies with two electrodes in which there were epidural electrodes, screw electrodes, which gives a good sort of distinction between the frontal and the temporal locations. And that led to these data that Khalil mentioned, indicating that there's a reproducible increase in resting gamma power, again here as a ratio of knockout to wild type in both the auditory and the frontal cortex in these mice, and that there's also this reproducible reduction in phase locking to the chirp stimuli. So that was something Khalil's already gone over, but that was a stimulus to us. And then in discussions with our human colleagues, we were thinking, what is the best translational opportunity here to try to make the mouse data more congruent with the human data? And it came back to a few things. First of all, there were other data in not only that the increased resting gamma power and the chirp synchronization were abnormal in the humans with Fragile X, but there also were these data about coupling and coherence. So, and I don't have time to go through those studies. I'm happy to communicate by my email will be on the last slide, and I'm happy to share all this information. But John Sweeney and Craig Erickson and Lauren Etheridge and others, Jun Wang, did these studies in which they looked at actually band-to-band coupling. So if you actually look at the amplitude coupling between frequency bands, in this case, theta to gamma, and also the spread of gamma activity across the cortex, so-called spread of coherent gamma, there's various derived electrophysiological measures that they could do in the human EEG, given the fact that they had 128 leads. And that was not possible for us to do at the outset in the mouse EEG. And we're still working on trying to do that, as I'll come to. But the idea was that there's these other EEG measures that were of particular importance and have become of particular importance in the human EEG work, such as coupling, coherence, and spread of activity, that are really best assessed with a multi-channel EEG data. So in the human data, they're able to do this because they have basically a dense array EEG over the surface of the brain. It's over the scalp, to be exact, with 128 leads. And our initial mouse data, although it generated a lot of great data, as Khalil mentioned, is really just 2 screw electrodes. So it wasn't really so congruent with the human situation. So one sort of obvious opportunity here, which we tried to seize on, and that's the point of this talk, is that if we were able to basically create a multi-channel EEG in a mouse on either the scalp or the, and we turned out to do it on the skull, as I'll show you in a minute, that could increase the correlation power, and better cortical sampling may reveal new findings. It may allow us to actually do some of these derived EEG parameters in the mouse that were not available before. So that's the sort of the basic rationale for developing a multi-electrode array system in the mouse. And I have to acknowledge the constant discussions of our Fragile X Center group that led to these discussions, and then also this was supported by the National Institutes of Health in a supplement to our center grant that actually enabled purchase of the MEA system, which is obviously an expensive proposition. So I'll just talk about a little bit about why do this, and hopefully it's already clear based upon Khalil's great presentation, but really, we want some objective physiological outcome measures. One of the issues that I understand, of course, in the Fragile X field has been you want to be able to hang your hat on something as a biomarker. And so, of course, EEG with auditory hyperexcitability is objective. And then the fact that, as Cleo mentioned, there are these parallel alterations in EEG in mice and humans was a very big stimulus to trying to develop this further. In our initial studies, we did some studies under light anesthesia with the mouse, and that's, of course, a big confound for EEG analysis. So we wanted to develop a system where the mice are awake and freely moving and completely avoiding any confounds of anesthesia. So all of the studies I'm going to tell you are done with the mice awake and freely moving at least several days after surgical implantation of the electrode array. And then, of course, and Cleo mentioned this too, this was also very important, the actual data collection, data processing, the actual exact nature of the auditory stimuli and the analysis are very consistently done according to the same methods used in the human study so that we can generate parallel data streams. The hardware involved here, there's a variety of systems out there, and we settled on using this system, which is from a company called NeuroNexus. It's in our recent papers, but basically it's a flexible MEMS array. This is polymer MEMS technology, which basically has this microprinted EEG electrodes. These are little platinum electrodes, 500 micron diameter sites. And you can basically see how it's organized, where bregma, and I'll show you the mouse head picture in a minute, will be always consistently where that red dot is. And that's, of course, critical so that we can compare anatomical localization across different mice and, of course, the appropriate mouse strain and age match. You have multiple options for channel count. We ended up settling on a 30-channel probe for reasons based on anatomy. The other components of the system that are critical to generating interpretable data from a multi-electrode array system is a head stage. A head stage is basically an analog to digital converter, which digitizes the neural signal, the microvoltages that you get at the level of the head. And that's critical because it minimizes noise and movement artifact. Movement of the head stage relative to the head is also one of the huge potential confounds. And you'll see how we have eliminated that in the methods. A commutator is also important. That allows for uninterrupted recording or stimulation. In our case, the stimulation has been auditory. But I'll just throw out there to those who may be interested that this doesn't have to be limited to the auditory realm. You know, visual evoked potential could be done with occipital recordings with multi-electrode arrays. And there are those out there, notably one group in Germany that has done VEPs in arrays. And so it's not limited to any particular system, but of course, based upon our collaborations with Khalil and the fact that the auditory system was set up at UC Riverside, that's of course what we've done. The idea, of course, is that the commutator can swivel and rotate and move as the animal is moving. So to show you how these methods were developed, and by the way, this was really it's been just an amazing experience with my friend and colleague, Carrie Jonak, who joined the lab a few years ago and really took this on and has made huge progress in this technology. You take the electrode, which basically you can think of it as a thin film, and it's very fragile. It actually, then the brain surface is prepared. We have anchor screws, and the dot there is on Bregma. Many of you, of course, recognize the anatomy there quite well. We have this is the electrode then on the surface, the multi-electrode array on the surface of the mouse skull. So this is skull surface recording. This does not need burr holes. It does not actually penetrate the cortex or anything. And we can come back to other potential recording paradigms that can be combined with this at the end of the talk in future directions. But basically, that's what we've done. So we put a multi-electrode array on the surface. And then one of the issues, which is really Carrie put huge effort into over time, was this question of reusability. There's a big sort of financial or issue with, especially in the earlier generation systems, where these electrodes and the systems were prohibitively expensive. Now you get the system and the electrode still is about $600 per electrode. So we meaning Carrie, developed methods to reuse these electrodes over several times, at least 5 or 6 times per electrode, such as putting Teflon over the electrode and securing it in place. And there's a lot of details there that are in our publications. And if you want to implement this yourselves, we're very happy to be in touch about this. The other thing about the head stage itself has to be very carefully connected. And this is a post that she developed to be able to do that. The whole thing is then acrylic in place. And she's able to, at the end of recording from a mouse, which can be recorded at least for a few weeks now, and that enables some more chronic studies with MEA in place, can go back and see that the electrode didn't move, which is an important verification at the end of the experiment, of course, to include the data, and then, of course, reuse the electrode. So those are methods that are published in this paper, but we've been developing in order to make this sort of feasible for larger scale, drug trials. Basically, we wanted to be able to do this at a scale where we could really test this system out. First of all, establishing the baseline, but then doing drug studies. So this is just a video to show the mouse moving. This is at least several days after surgery. The mouse is, back to normal activity, and you can see the commutator moving as the mouse is moving. The head stage is carefully implanted so that the mouse is able to move. Now, so of course, the key to all EEG data is signal to noise, as everyone knows. And I've been dealing with signal to noise on EEG for most of my career. But based on the ideal recordings where, this ideal method that, I don't know about ideal, based on the revising of methods so that Carrie is able to generate very good noise, very low noise EEG. And these are raw traces, so this is not filtered for artifacts. So she's getting really good recordings. Now, one of the things got me excited early on about this is that even if you just look at the 30-channel raw EEG on the wild type and the 30-channel raw EEG and the knockout, and this is just freely behaving EEG, free running EEG, nothing, no stimulus at this point. You can see quite a remarkable difference here. And even my 9-year-old son can see that these don't look the same. He says this one looks more squiggly. So basically, obviously we do a lot of post-processing analysis, but I wanted you to see that we really do see a big difference in the EEGs in the knockout and wild type at baseline. And this is related to increased power in the knockout EEG. So then the question becomes, you start generating these data, and how do you analyze these data? And that's still a work in progress, but I'll show you what we've done so far. This again has the picture of the probe on the mouse skull with, of course, the bregma very carefully located at the site of the contact here. And each channel is carefully numbered. And then, of course, you have to follow that through the post-processing, the software, and all of those things. Carrie did a lot of work to work with on the software as well. But then we grouped them just empirically, and so we could analyze these things into right and left frontal, right and left temporal, and right and left medial. And I'm not saying that's perfectly anatomical to various lobes of the brain in the mouse. It's a different structure than human. It's lissencephalic. But at least it gives us the opportunity to analyze different regions in a region-specific way. And we can still go back and look at individual electrode contacts. So then basically we wanted to implement these methods into simply reproducing, if we could, the results that were done with Johnny in the 2-electrode system. In other words, the resting EEG and the chirp, and also auditory ERPs. So this shows that basically they're recovering, so there's no anesthesia on board at the time of these recordings. They're brought into the recording chamber and connected to the head staging commutator, and 15 to 20 minutes or so habituating, so that they basically want to be in a normal state of activity. And these data, and this in our recent publication, is based upon 10 wild type and 11 knockout mice. We get about 5 minutes of resting EEG with no sound, and then the auditory stimuli are presented, both ERP and chirp stimuli. The resting data, and this is again presented as ratio of knockout to wild type, where the knockout is black and the wild type is white, demonstrate that there are some region-specific differences, but across the board, there's this increase in gamma power in the knockout, similar to what we saw with the 2-electrode data. What's interesting is we get more, of course, region-specific data, both left versus right, and we get more frontal versus temporal information. Notably here, you'll see that across the board in the frontal region, there appears to be a more broad, across different frequency bands, increase in power as compared to, for example, the temporal region. We also looked at ERPs, and this is an averaged ERP across many stimulus presentations. And basically, we reproduced the what had been seen in multiple previous studies about the temporal N1 component being increased, and also left and right frontal and medial components we can analyze separately. That's shown here. It's the details are in the recent paper that Carrie published. But basically, as Khalil showed for Johnny's data, the 2-electrode data, we are seeing the significant increase in N1 amplitudes. This here on this side is amplitude, and this is latency. So we didn't see much difference, or if any, in latency to these different components of the auditory signal. More a difference in amplitude. So those are also potential outcome measures for auditory stimuli. Most impressively to us was that we were able to reproduce the chirp deficit. In other words, this interesting 2-second auditory chirp stimulus that Khalil described very well. In the wild type, you get this really nice chirp response where there's good fidelity to the temporal stimuli, temporally modulated auditory stimuli here. And over those 2 seconds, it's weaker in the knockout. And the difference, knockout minus wild type, is shown in these type of panels. So I know there's a lot of data on this slide because we try to include all these different regions. But if you look at, for example, left and right frontal regions, you see that there are deficits in both the right and left frontal that look pretty similar here in the knockout minus wild type. And in this gamma range, these black areas are the statistically significant differences. And you, again, just like Khalil showed, you see this gamma range abnormality in chirp synchronization in the knockout mice as assessed by a surface MEA. So it's further confirmation in our minds that first of all, that the 2 electrode mouse data that were generated are indeed validated, but also that we can reproduce this with an MEA system. There's a quite impressive phenotype in what we're calling the medial region of the brain as well, where you can see quite an impressive change in the chirp synchronization, even across into high gamma areas as well. And then also in the temporal region, you see a very significant change in between knockout and wild type. And this has been our most reproducible phenotype of decreased cortical phase coherence to these stimuli. So to summarize where we were at that point, which is really the recent paper that we published in early 2020, we've basically put a lot of effort, and this is all Carrie's work, to basically compare knockout and wild type mice. And basically, we found indeed that there was this enhanced resting state gamma, and that was low gamma across the board, but of course other frequency bands in the frontal area as well. Increased ERP amplitudes, which is similar, particularly the N1 amplitude, to what has been seen before. But really our most sort of what we consider robust phenotype is this remarkable decrease in the chirp phase locking that Khalil described as well. So these results are very similar, which gives us some confirmation of the 2 electrode recording system that was done in our previous work, but provide more spatial information and then potentially statistical power. I'll mention that the N that we had in these groups are 10 and 11, that we had to go to much higher N in many of the studies with 2 electrodes to get good statistical data. So that's sort of an important issue as well, that it gives us a little bit more statistical power just based on the number of electrodes involved. And most importantly, that results that we are seeing with this MEA system are similar to the results seen in Fragile X patients, notably the gamma power change at baseline, the increased ERP amplitudes, and the decreased chirp response. Okay, well, we wanted to then go forward, and the rest of the data that I'm going to show you are as yet unpublished, and we're involved in multiple testing at this point of candidate drugs to see what they do to these EEG biomarkers, and that's one huge thing that we're involved in, both current and future directions. We were influenced by, as a test of this, Siegel Lab Sinclair et al. Paper in 2017. We were very influenced by this because they used the racemic baclofen, which is this GABA B agonist, and they found that it actually does reduce in a dose-dependent way these 2 doses they tested, both the EEG, but they reduced the EEG gamma power in the Fragile X knockout mice. So we saw this as a nice test to see what acute baclofen and also chronic baclofen might do to the multi-electrode array EEG power and auditory chirp response. So what we've done, and Carrie's done these studies as well, is administering a low dosage and a high dosage of the baclofen, and these are the exact same dosage as in the Sinclair paper, and N of 10 in each group for each of these dosages. And this is the acute single dose study where basically we get pre-drug baselines for each mouse as we've done in all of the knockout and wild type studies to date. And then we give the racemic baclofen, and then 1 hour post, chosen to be at a time point when the acute baclofen dose is hopefully at its maximum. We do resting EEG and up chirp, and these are the details are that the up chirp is actually 300 trains, and it's all average to get the data that you see. So what we've see is that basically now we're looking at pre and post baclofen. So this is a little bit differently organized to the data before. The left is wild type and the right is knockout. So I know there's a huge amount of data on this slide, but just to summarize what we've seen so far, in the wild type, when you give 2.5 milligram per kilogram baclofen, 1 hour later, you see actually quite a large change in the low frequency of the EEG, namely an increase in delta and theta power. And delta power is the most consistent across brain areas. You do see that also similarly in the knockout, that there's this increase in delta power, although not much change in other frequencies at this particular dose. So this is what we classically refer to in EEG literature as EEG slowing. EEG slowing basically means that it's almost like a partial anesthetic effect of a drug in many cases, where the cortex is slowed down, and meaning that the power in the delta range, the low frequency power, is increased. So that's more or less what we're seeing here with the acute 2.5 milligram per kilogram baclofen. And then at this dose, the ITPC, the intertrial phase coherence, or the chirp synchronization response, is not significantly changed in either the wild type or the knockout, although there's some trends here in certain areas of the knockout that look like it might slightly improve the chirp stimulus response. But that's at 2.5 milligram per kilogram. When we go up to the higher dose of baclofen at 5 milligram per kilogram, and I should mention, this is of interest to drug development, I think, this dose of baclofen is sedating to the mice. So that obviously becomes an interesting point behaviorally in terms of what we're doing to the brain. But basically, there's a marked difference in now the EEG is markedly suppressed at this dose. So you see there's a very different effect at this dose compared to the other dose. In the wild type, you have a marked suppression of EEG power. So post versus pre, there's much reduced power really across the brain. So it's really what you would call anesthetizing the brain in that sense, that the EEG power is markedly suppressed. In the knockout, it's interesting that it doesn't seem to suppress as much at this dose. So that's also an interesting knockout wild type difference. But there is this trend towards suppression of gamma power in the knockout at this dose. What we found remarkable is that at this higher dose of baclofen, we are seeing this remarkable improvement in chirp synchronization. This is seen in both the wild type and in the knockout. So this is really restoring, if you will, the knockout chirp response to something that's, or I'd say improving it to something much better than it was previously. Now, what this means in terms of behavioral relevance and everything is speculation, but we certainly see this remarkable dose-dependent effect on the chirp response here, given the higher dose of baclofen. So to summarize where we are, and then I'll talk about where we're going with this. We have worked hard, and this has all been Carrie's work over the past few years, to implement this 30-channel surface multi-electrode EEG analysis in wild-type and Fragile X knockout mice, and done multiple studies to try to implement the technical aspects of it and then make it reproducible. And what's clear at this point is we now have robust phenotypes of increased resting EEG power, increased ERP amplitudes, and reduced auditory chirp response or phase coherence in the knockout mice. And I probably should have mentioned, these are all adult knockout mice, age matched to the control mice. We've seen some interesting changes in EEG in the multi-electrode array EEG with acute single-dose baclofen administration, namely dose-dependent suppression of EEG power, but interestingly, this increase in phase locking to chirp stimuli. And we have this sort of gamma noise hypothesis that Khalil kind of referred to, that this knockout brain is running along with this gamma noise, with this high amount of gamma noise, and maybe quieting the noise with the baclofen enables the synchronization of the cortical neurons to chirp in a better way. That's a speculative hypothesis, but it fits with the idea that suppressing gamma in the knockout will improve chirp. One thing that we're working on currently, and we don't have the final data yet, but we're in the process of doing this, is we want to also be able to use this system to compare acute administration of drugs to chronic administration of drugs. We're very aware of the information in the Fragile X literature, notably with the mGlu-R drugs, that there appears to be this tolerance problem. So we want to be able to test drugs both acutely and chronically. And chronically, of course, means one thing in a human and another thing in a mouse, but we are at least able at this point to do several weeks of recording with these MEAs implanted. So we're currently involved in comparing the one-day baclofen data versus 2-week data to look at chronic effects versus acute effects. And that should enable us to determine, is there some tolerance to this drug or any particular drug that we test in the system to the EEG parameters. So I want to just sort of describe our future directions to this work. It's highly collaborative. We were lucky to be involved with the expert auditory neurophysiology of Khalil and his lab in order to be able to implement this, and also the Fragile X Center group at UCR and also across to Cincinnati and Texas. We want to test candidate drugs. We see ourselves as sort of a translational link in this whole process. So we're very excited to work with, Mike and FRAXA and anyone across the world who wants to test drugs and do so in a way that we can try to determine the EEG biomarker engagement, meaning like, does it affect gamma? Does it affect chirp? Does it affect it acutely? Does it affect it chronically? And to be able to assess, tolerance effects. I haven't mentioned anything about behavior, but of course we we like to collaborate with people who are doing behavior analysis so that we can be the EEG component of a drug analysis potentially here as well in our lab. The other thing we're doing, and I'll just mention a few other things, then I'll stop. We really want to derive, as I mentioned before, these other EEG biomarkers. So of course we've gone to resting EEG power, ERPs, and chirp. But we really want to, and then we're in the process of doing this in collaboration with the University of Cincinnati group, to get coherence and band-to-band coupling, such as theta-gamma coupling, alpha-gamma coupling, which has been shown to be abnormal in Fragile X patients by Lauren Etheridge and others. Phase amplitude coupling, which is a very important cognitive measure for memory in many studies in humans. Principal component analysis, which is currently underway by Ernie Pedapati at University of Cincinnati on our mouse data. And also something called debiased weighted phase lag index, which is a measure of spread of cortical synchronization. This has also been seen to be abnormal in Fragile X EEG in patients. So we want to get that in the mouse if possible. And so this part is in progress, but we see this as a potential for developing hopefully even more robust biomarkers that may be more behaviorally relevant or cognitively relevant for translational therapeutics. Next, we also are open to looking at other ASD models, and we are in some discussions about that because I think it's potentially to look at other EEG phenotypes that may be useful in other autism spectrum disorder models. And it'll be interesting to compare, for example, Fragile X knockout mice to other models. And of course, we've also considered the Fragile X knockout rat, although we haven't done that too. So it's another interesting discussion. Combining surface with depth MEA analysis together with Khalil and his expertise in auditory neurophysiology in the cortex and his single unit recording data, it would be of great interest to combine a depth multi-electrode array, which now we have the opportunity to record every 20, 30 microns or so in the cortex with depth MEA. And doing that in conjunction with the surface MEA is something that we want to do technically so that we can further look at cortical physiology and understand that a little bit better in Fragile X, both at baseline and in response to auditory stimuli. And of course, depth MEA could be done at any point in the cortex. It doesn't have to be limited to the auditory system for those who are interested. Overall, the whole this work is explicitly translational, that we want to try to provide a bridge between these EEG biomarkers that are seen in humans and try to identify more in mice that can be used for outcome measure analysis. So I just want to say the people who actually did the work. So this picture here is the Fragile X Collaborative Research Group, and you see Mike there, who attended the meeting, and all of the people from Cincinnati and Texas. And then the people that actually did the work I've talked about it. Carrie did the multi-electrode array development and work that I've shown. And of course, Johnny did the 2-electrode work. And I'm indebted to Khalil, obviously, for all the auditory physiology. So I think I'll stop there. And please email me with any questions that we can address during this talk at my email address there. Thank you.
Really great talk, Devin. And I know we've got a lot of interest in the presentations and a lot of questions. So I'll start at the top, and just to read these into the record. Johnny asks, why is it important that we can identify differences in wild type versus Fragile X mice? It's all about the phenotypes. Anyone want to comment on that?
Well, I mean, it's to identify the phenotypes that we can then test therapeutic agents against. And also, if the differences are what are going to allow us to get into the brain to understand what the underlying circuit mechanisms might be. So,
Shika asks, P14 and P21 in mice translate to what ages in humans? Is a similar difference observed in humans? Electrical stimulation as an intervention to attenuate the extended sensory response to stimuli in Fragile X individuals. Your thoughts, pros, cons?
So that's a tough question, because I think different parts of the brain sort of develop with different trajectories in relation to how human brain develops. So the way I think about it, and the way our group thinks about it, is that the P14, P21 window in the mouse is sort of the critical window period for auditory system development in the mouse. And if we look at human auditory system development, that's probably extended in the first 6 years of life, or first 6 to 7 years of life, is where a lot of the critical period plasticity is happening in the human auditory system. So I think the P14 to P21 in the mouse might correspond to less than 6 to 7 years of age in humans. Whether similar differences in terms of development of hypersensitivity has been observed in humans, I don't know the answer to that question, but I know Lauren Etheridge is starting to record EEGs from younger and younger children, and hopefully her data will shed some light on that pretty soon. And for the last part of the question, whether electrical stimulation can be useful, again, I don't know the answer to that one, but I do know that Ernie Pedapati at the Cincinnati Children's Hospital, one of our collaborators, is using more of a magnetic stimulation approach, TMS sort of approach, to see if that provides any benefit for hypersensitivity. Not electrical stimulation, as you're asking, but certainly people are thinking about stimulating the brain in a way. Yeah, so that's where it is right now.
And I guess I would just add a bit of calibration in that the knockout mouse experiences a peak in seizure susceptibility, at least for audiogenic seizures, at 3 to 4 weeks of age. Mm-hmm. And in humans, generally speaking, the peak seizure susceptibility is between 4 years and about 9 to 10 years of age. Mm-hmm. So if that gives you a rough correspondence of how the mice and humans stack up, at least in terms of seizure susceptibility.
Yeah, I do think there's a lot more data that needs to be obtained in younger children with EEGs.
Danny Graziano asks, to piggyback on the previous question, how long after P21 does that spiking phenotype and the perineuronal network and MMP9 abnormalities last?
Yeah, so the MMP9 increase is seen even at P40. The spiking abnormalities are seen in the adults, as we've seen. So between P21 and the adult, it's present. The PNN abnormality is not, it doesn't really follow this this trajectory. I think the PNN abnormalities, the PNNs become more normal by P30 in that age, in that time frame. So a lot of these seems to be setting up a trajectory, and then a lot of the phenotypes disappear. I think that seems to be more of a rule than an exception in Fragile X research, is that many of the phenotypes seem to be transiently present, and then they disappear and then reappear. And I think there's a lot of compensation going on, that some phenotypes are seen for a brief amount of time, disrupts development during that time, and then disappears, only to affect something else later on. And that's one of the complexity of looking at any developmental disorder, is how much of what we're seeing at any given age is because of the genetic reason, as opposed to the experiential compensation that happens to compensate for the genetic mutation. That's a tough problem.
Carol Wilkinson asks, how do mice EEG abnormalities relate to the behavioral phenotype, cognitive deficits, behavioral responses to sounds, hyperactivity, etc.?
I think that's a great question, Carol. I don't have a great answer from our data because we don't do behavior in our lab, but I can say, I recommend to everyone look at that Sinclair et al. Paper from Siegel's lab, because they actually tried to do that with the baclofen. And they looked in individual mice. If baclofen normalized gamma, did that improve behavior? And they did anxiety-related behavior, and they did working memory. And that's probably the best paper. There's other papers, there's plenty of papers, but where they showed that in the mice that had more normalization of gamma, those were the mice that did better in terms of improvement in anxiety-related behaviors and in working memory. So they had, like you always have in vivo studies, you have the spectrum of response, when you give a drug, and you have this in vivo variability that's inescapable. But they were able to sort of mouse by mouse, so to speak, correlate the fact that gamma oscillation power reduction by baclofen acutely improved anxiety and improved working memory. It didn't improve every memory task, for example, location memory and things. So they did some other tests where it didn't help. So I think for every drug, you're going to have that type of question, that if it improves certain EEG abnormalities, can you show it also to improve behavior? But I think that the correlation, Carol's question, gets to the question of, what do we predict if we normalize gamma power? And that, I think, still is a good open question. But at least that one study suggests that both anxiety and but it could be unrelated. GABA B agonism by baclofen could normalize anxiety through mechanisms other than the fact that it also has an effect on gamma. So we have to be a little bit careful about causation. But anyway, at least in that study, there's a correlation between reducing gamma and improving anxiety and memory.
And to add to that, I think the human work also showed correlations in sensory hypersensitivity and the SCQ questionnaire with the elevated gamma power in children with Fragile X, or adolescents with Fragile X.
Okay, Matthias asks, in addition to parvalbumin, did you also look at the effects of other subclasses of inhibitory interneurons, such as somatostatin?
Yeah, so these are studies that we're setting up to do. We are starting to devise mechanisms to activate or inactivate either PV cells or SOM cells to get more into the function of each of these subclasses. So at this point, the answer to that question is no, but hopefully within a year, we should be able to give you a response for that.
Another question from Carol Wilkinson. What region of the human brain do the mouse medial electrodes generally map onto?
Well, it's another great question from Carol. What can I tell you? I don't have a great answer to that one because there's so much difference. I mean, I don't mean to sound hand-waving, but you have a lissencephalic brain in the mouse, and there is structure to it. Obviously, you have somatosensory and motor strips, just like you have in the human. And that's approximately where we are, just lateral to the midline with these medial electrodes, which are showing these huge chirp differences in the somatosensory motor area. However, That, of course, with the homunculus, even in a mouse, there's a homunculus that in humans is better defined, as we know from Wilder Penfield and others. But the medial region there is obviously not the auditory cortex itself, because the auditory cortex itself, as we all know, is temporal. A1 is temporal, and it's not frontal. So some portion of that posterior medial area is still frontal, and some of it's probably parietal lobe. But it probably encompasses the medial part of the motor and somatosensory area in the human, which would involve the lower extremities. But anyway, that's a somewhat hand waving because we don't really know. And I think we have to be a little cautious because the actual electrical recordings from humans, still they have much more data still, 128 versus 30. But also we're talking about very big differences in space. So the interelectrode distances in humans are still larger, even though they have more of them because the human brain is so much bigger. So we're a little bit cautious about trying to map the brains together. That being said, we do want to do studies of mouse MRI, and we haven't done it yet, but this is just in the discussion phase, where we can then map these electrodes by distance from bregma to the surface and figure out exactly what mouse brain structure is underneath. So we, of course, have mouse brain atlases that we can look at, and it shows basically what I just said, that it's in the medial somatosensory motor area, but it doesn't really help in terms of more localization than that. And then mapping it to human, very difficult problem. So I don't know if that was what you expected for an answer, but we don't really have a great idea. Although in general, the homunculus is thought to be similar.
Johnny has another big question. What is the difference between treating symptoms versus specific disease treatment?
Well, I mean,
and how does it apply to this work? I guess I just append that little comment.
Yeah, I mean, we'd like to treat the disease, but at this point, all we're doing is treating specific symptoms. The main, in fact, the overarching sort of goal of some of the work that we do here is to see if actually treating some symptoms early on in life does have a broad benefit later on. And that's the main thrust of the work that we're moving forward with, is to see if we can treat sensory symptoms early on, and that reduces anxiety and cognitive deficits later on. And that's a key goal. Again, we're not treating the disease, we're treating symptoms.
I don't think that's a bad thing. I think, with diabetes, you're controlling blood sugar, but you're not curing people with diabetes. So, I mean, if we can do something similar with the brain, at least, we're engaged. I think engaging the right cortical circuits and the right mechanisms is a more approximate goal. Obviously, there are gene therapy studies for, as Mike knows much better than I do, for Fragile X, and we would love to also, be involved in those to see what for example, gene therapy does to EEG. So, when it comes to restoring the actual, genetic basis back to normal, sure, but we could also use these as maybe outcome measures.
Tanishka asks, what does enhanced gamma power mean? Does it reflect excessive firing of a particular population of neurons or increased firing in general?
Yeah, that's a great question. I mean, we use gamma sort of loosely in a way in the literature. There are different bands of gamma. So there is the low gamma, and then there's the high gamma, which can be generated by different mechanisms. So what we know now, it seems like pretty clearly, is that the high gamma frequencies are mostly related to enhanced firing in general, right? It's not associated with any specific neuron. It's just cortical activation. So anything above 80 hertz, 90 hertz gamma seems to be simply associated with enhanced noisy firing. Right? The lower frequency, more narrowband oscillations that are seen between, say, 30 and 60 hertz, that is related to several models now that related back to the firing of the PV cells, and that's still that's a topic that is still being debated in terms of, there was a previous question about somatostatin. So what extent do somatostatin neurons contribute? To what extent do PV cells contribute? So the key is to distinguish what are narrowband oscillations from what are broadband oscillations, and to distinguish the low frequency versus high frequency oscillations. And one thing for clear is that the really high frequency oscillations in the 75, 80 hertz are associated with increased firing, increased spiking of just cortex, not necessarily any neuron, just increased firing. That's the best response at this point.
Okay, Julie Charlton asks, can FMR1 mice with the multi-electrode array installed also be assessed for behaviors? And I would just add to that, has anybody ever thought of just counting revolutions on that commutator?
Yeah, that's a good point. Well, Julie, great question. So yes, we want to do that. We think so. I think it's going to be somewhat limited to the physical nature of the behavior being assessed. So complex memory task where a mouse has to go through various places, you can imagine ways in which, as long as that commutator can somehow be suspended from, it gets a little complex, but in some type of, I don't know, radial arm maze or something, as long as the top is open, you could imagine potentially connecting the mouse. So we have not done that, but those are really interesting discussions. Certainly, we know from the standpoint of just how long the electrodes can stay on, we know it's several weeks at least now. So we're pushing that out to see these more chronic drug studies. But you're right, with behavioral analyses, we'd like to see what we could ask the mice to do while being connected. And basic behaviors in the cage, like nesting behavior, marble burying, these types of things that have been done. We've talked about doing them. We haven't done them. We're not a behavioral lab per se, but we have talked to, just for example, we've talked to Mike Ragazzino, who is a behavioral, really an expert in behavior. This is particularly something called probabilistic reversal learning that he's developed as a test that distinguishes knockout and wild-type mice in Fragile X. And so we have as a proposal going forward to work with him to set that up at UCR together with Khalil and my lab and try to test these mice before and after, maybe even during, but before and after probabilistic reversal learning to look at the effects on EEG of behaviors. So it's a great question. It's a huge topic, but I think the answer is yes, as long as the EEG recordings can still maintain good fidelity during the behavior.
So Mike, as I mentioned earlier, I have to leave at 9:30 to teach. So I'm going to sign off at this point, and I really thank all of you and FRAXA for the opportunity to do this. I'm also very happy to answer any questions via email, so just find me, and then I'm happy to respond. There are some great questions towards the end that I'm not able to get to, but I'm happy to answer over email. So thank you so much. I have to rush to teach. Sorry.
Thank you, Khalil.
I'll
I guess we have time for a couple more with Devin, perhaps.
Sure.
From Durgesh.
Thanks, Leo.
Bye, Devin.
Thank you.
Durgesh asks, great talks. Isn't it worth normalizing the knockout and wild type individual waveform data to total EEG power first, and then compare the knockout and wild type signals for waveforms rather than comparing it directly? And is the elevated gamma constant during the day and nighttime? I'm asking if you've checked any correlation with REM sleep cycle.
Hi, Durgesh. Those are fantastic questions. Thank you. First of all, on this issue of relative versus absolute EEG power, you make a very good question or point. So what we've done so far is basically, as you know, absolute EEG power, total EEG power, and that's where we've seen these types of phenotypes. Once you normalize to the total EEG power and then look at relative power across various frequency bands, we haven't done that in detail, but we can do that. That's relative EEG power. It's not clear to me that that is more valid or less valid than it's just a different parameter than absolute EEG power. In most of the human data, it appears they're going with the absolute. So that's where we've been. But I think you're right. It's worth comparing that as well, because it may be the case, for example, that a given drug may affect the balance between, say, theta and gamma power, but not affect overall power. So those are the types of things that we should look at as more of a relative ratio. And we're trying to do this, and as I say, in collaboration with the Cincinnati group, to be able to hopefully soon spit out a lot more data faster through this EEG pipeline analysis that I haven't had a chance to discuss, but it's basically to try to get all these parameters, both relative, absolute power, principal component analyses, theta gamma coupling, all of these types of things will hopefully be implemented soon. But you're right about that. And then the other one is the day and nighttime and the circadian rhythms. We have not assessed. I think it's a great question. In my first job ever as a scientist, I work as an undergrad at Harvard in Alan Hobson's lab, we did REM sleep research. So I really appreciate you bringing this up. We have not done any circadian rhythm analyses, any sleep cycle analyses. These have all been awake recordings with the mice freely behaving, as you saw. So we don't know the answer to your question, and maybe we can talk about how to do that. But so we don't know the answer to that. I think the circadian rhythms are clearly disrupted in Fragile X. There's other papers on that, and we just don't know what the MEA does in sleep. We just don't know the answer. But it's a great question.
Okay, I think we got at least one more from Arjun. Says, did you ever see intra or intervariability in FMR1 knockout data compared to wild type? If yes, do you consider them as another feature of Fragile X? So I assume he means variability within the Fragile X knockouts.
Right. So we certainly, of course, like any in vivo study of anything, you have some variability. So the question is there overlap, and what's the variability within the knockout versus the wild type groups? Now, I recognize that many, and Mike, of course, knows this much better than I do. The actual Fragile X knockout model is not exactly an accurate model of human disease for obvious reasons. There's various, it's a complete knockout. So we have what you might call the, lowest possible variability because it's a knockout. And we use littermate controls for our most recent studies, which is another important issue that reduces variability. When you start to get outbred mice and you purchase them from various places and stuff, you'll be interested to know that the EEG looks very different. And one thing I should mention in this context is the C57 versus FVB background of the mice can make a huge difference in the resting EEG. So you have to compare apples to apples. That being said, we really have seen, I think what's been most variable is the ERPs that kind of can be all over the place, but we certainly have this remarkable chirp synchronization problem that we see really robustly in the knockouts. And I think that relates really quickly to the fact that, when you're recording amplitudes of things versus temporal synchronization, the temporal synchronization response of the EEG is more indicative of what the brain is actually doing. The amplitudes also can be more artifactually changed by precise positioning of electrodes or blood or scar tissue or things. So just to be clear, the data are consistent, but I think we're getting the most robust data in the chirp, and that sort of really separates the groups. I hope that answers. If not, please email me more questions about that.
Great. Well, I think that concludes our Q&A session, and that concludes our webinar for today. I just wanted to thank our speakers once again, Dr. Devin Binder and Dr. Khalil Razak from UC Riverside. Great presentations, and I know there's a phenomenal amount of interest in the work that you guys are doing.
Well, thank you, Mike. Thank you, FRAXA. Thank you, Katie. Thanks, everybody. And like I say, please email me or Khalil. We like to collaborate within the community, and it's really great discussion today. So thanks again.
Awesome, and thanks to Eric Wellen for keeping everything working.
Thanks Eric.
No
Have a great
problem.
day, everyone. Thank you.